Hi, my name is Kyle McCary. As she said, I'm from Texas A&M University, and I'm going to introduce our speaker here. So Dr. Pan is a research physicist at DEVCOM Army Research Laboratory. He's been there since 2009. He's also an elected ARL fellow, who is one of the most eminent scientists or engineers in ARL. He owns a PhD from East China Normal University from 1993. His research is focused on inventing and developing sensor systems for real-time in situ bio and chem aerosol detection, mainly based on single particle fluorescence spectroscopy, two-dimensional angular optical scattering, and Raman spectroscopy. He published in over 150 referee journal papers and 3 book chapters, was awarded 11 U.S. patents, had over 5,000 citations from other researchers. He has successfully completed more than 20 research projects as a PI or co-PI. The title of his presentation is Real-Time Detection of Single Bioaerosol Particles via Differential Circular Polarization Scattering. And you are welcome to drop questions in the chat as we go, as we will have a Q&A session at the end of his talk. And so now I'm going to pass it over to Dr. Pan. Dr. Pan, you're muted, just to let you know.
Is it not good?
Yes, you're good now.
Okay. Strange. It's work before it's muted. You will see that here, our young colleague is much smarter than me. Sit here, I feel more confident. Okay, then can you see the screen?
Yes, we can. You're good to go.
Okay, then it's good. So let's start. So the title is Real-Time Detection of Single Bioaerosol Particles using the Differential Circle Polarization Scattering. So I have worked in this field more than 20 years and started at a year, then joined the AIL. Those years, people, and I think the started to get hot for this topic is 1996, 1998. That's the first paper for user fluorescence to biodetection. So biodetection actually have been existed for a long time. So that time get hot is start doing the aerosol detection and based on single particle point detector. So like last couple years for the COVID, and the various people have to collect the sample, then to use a PCR or early other method for the biochemical method, then take a long time. And of course, it's much more accurate, precise, that can identify the biomaterial. So for the first method, and people generally use the elastic light scattering, fluorescence, Raman, and breakdown spectroscopy and mass spectroscopy, then some absorption and also the LiDAR systems. So for this different advantage and disadvantage. So for the single particle point detector, mostly based on the scattering and the fluorescence. And like the Raman and laser breakdown, they have more specific information, but they are generally more complicated and harder to run continually and from single particle. They generally deposit on the slider or some container, then to look one by one. So the most closest system, like Raman, is the system generally like the Battelle one, they deposit on the tape, then look at the Raman, then move the tape, then deposit, move. So it's kind of quarter and real time, but not real time. And this breakdown, the Livermore Lawrence Lab, the developer system, they can look at the single particle and mass spectroscopy and break down, then they go to see the property of the single particle. So that one then is generally is very expensive and big, and it's hard to analysis. Is somehow you need a little long time. So for the elastic scattering, you can get the size, shape, and the polarization, reflecting index. That can be quick, and some can be much smaller. And also the commercial one for the fluorescence, like from the TSI, and also from the droplet and measurement company. So those commercial one is get the fluorescence 2 bands and 3 bands by 1 wavelength or 2 wavelengths excitation. That's most common and commercial one. So, but however, for those system developed last 20 years, and they have some problems. For example, for the fluorescence, the most popular one, and you look at this, we noticed the problem, and about 10 years ago, so there is a campaign in the dog way, and a lot of optical system move to there and increase other system, and they release the isolates to the air, and of course it's the depleted, right? Then all the machine never see the particle. Then come to my mind, and we have a good discussion with Josh Santapia at Loch is in University of Nebraska Medical Center before he's at APL. And we think, okay, there may be 2 main problems. And the property we get from the laboratory, from the seminal material, is different from the real agent. That's one problem. The second problem is when the bioaerosol particle released to the air, the property changes quickly. Then we cannot measure it. So then come to and 2 big and project we worked on. So the first one is like the spectral, and we try to summarize all the possible wavelengths people use that. And so for the fluorescence for biological particle generally is at the deep UV from 260 to 280, and they get the amino acid get excited for the chocolate tyrosine, get the deep UV and get the strong fluorescence. Then the other part is from 340 to 360 for the NADH and riboflavin or the other molecule in the visible wavelengths. So the commercial and most laser people use is 260 and 280, and like a LED flash lamp, and then 350 and then 280 another, then other one is the 405 for the diode and blue or UV laser. So we use the 5 wavelengths. You can see here from the law, each law is from 263, 260, 273, and 280. Then the fourth law is the 350, and then the last one 405. Different wavelengths. The different column is the spectrum from different real bioagent and the simulant. The first column is the bacteria, and the second is the bacteria single. Then the third column is the virus simulant and the real and the agent. So you can see this spectrum is very broad band. They look very similar. So all have a very strong peak when you use the deep UV excitation. So the first 3 law, they have a strong peak around 330, or they have then a weak signal at the visible. And if you use the visible or blue excitation, as the 350 and the blue excitation, you can see they have a visible band, and they are very similar, but there are some strong, some weak. And that means that you use the different wavelengths and see different spectrum, but use the same wavelengths for the excitation, different material, their spectrum very similar. Of course, those depends on the condition, growth condition, or the concentration, a lot of small details. So it's very hard to distinguish between the different kind of bioaerosol particles. For example, even the bacteria, they are belong to the same. If they belong to the same bacteria, the E. coli and Bacillus subtilis or something like a different one, they hardly can tell the difference. So that's one problem. And the second problem is when the particle released to the air, we observed that they do have big change. And we have two review paper and quite a few paper published. So we can see their viability change and the particle, especially particle property, spatial property change. For example, look at the picture we presented here. So you can see where the left is the different and condition we put in the reaction chamber. So we put the bioaerosol into a rotating drum. Then we measure the single particle fluorescence spectra and excited about 263 and 351 nanometer laser. And the spectra change with the time. So the black is the initial spectra, then become blue and red and green. And you can see the first law. So the left is initially the UV peak is strong. When the time are half hour and every half hour the deep UV drop gradually and dramatically, and the visible band no big change, and the 351 excitation they barely have any change. You can see the very right column. The blue is the intensity of the visible band, and the black is the UV band. The red is the visible band by deep UV excitation. You can see all different we have the 5 example here, different condition we put into the chamber. Look at the exposure, the UV exposure, and the ozone, and some other and chemical reaction material in the generally and exist in the air. And you can see the most the drop there and the deep UV and the visible some no big changes, some decrease there. Look at the last spectra, you can see the visible even increased. And so you can see here from the black to the green is increased, the last law. So that's produce new material during the degradation process. So that means it's very complicated when the particle released to the air. And most of the system, they use the ratio of the UV and the visible of the total intensity of the bands to determine and what kind of biological particle you observed. And obviously, you can see here, it's not reliable at all. So how to solve this problem? Then we come to this idea, and we think that maybe the scattering have a better chance and all have a better property for very fast detection. Of course, this is for very real-time fast detection. It's like kind of the screen of the bioaerosol. It's not for identifier, keep in mind. So for the scattering, so people use the stock vectors to describe the latter property, the intensity, the linear polarization, the four polarization, and the circle polarization. You those can completely describe the latter intensity and the polarization status of a light. And once polarized light interact with a particle, and you get another the scattering, or the output also can use the stock vector to describe the property of the light. And the property of the interaction material or the single particle, you can describe by the Mueller matrix. That's the S11, S12, and that's the matrix. So each of them indicate a different property. For example, S11 is the total intensity, is generally the non-polarized scattering intensity of the particle after interaction with the laser and with the light. And the S14 is related to the circle polarization, like 1, 3, 4, or those, and 1, 2, and those related to the linear polarization, the depolarization. So we particularly pick up S14 and because that's a little easier to measure, and we measure the circle polarization by left circle polarization excitation and the right circle polarization excitation, and the intensity, and then differentiate them and normalize them. Then that's the normalize the matches of S14, and we call this CIDS, circle intensity differential scattering. And that's people reported, and in 1980s, quite a few people and did the experiment and the theoretical calculation. They said this can use it to look at the DNA, RNA, the helix structure, also some spiral and shape of the particle. They should be have singular, but for sphere and uniformly distributed molecule without early helix structure, then the singular should be 0. So look at this method that we pick up is because they have a lot of advantage compared to the fluorescent and other things. So you can use a visible wavelength, because for the fluorescent you always have to use the deeper UV or UV. So UV, the short wavelengths, they cost more and expensive and bigger and hard to get it. And for the visible, like 532, very common and very small, you can get enough power. Also, the elastic scattering has the largest optical cross section. Then a small laser, you get a strong enough signal. The fluorescence is weaker, and Raman even much weaker, is 10 to the -3 full order at least. And also, like the fluorescence, Raman is inelastic scattering process. You need a dispersed device like a spectrograph to look at the spectrum, and those is look at the signal at different wavelengths. But for the scattering, you look at the signal at the same wavelength at the illumination laser. So also this one can directly detect the DNA, RNA. So that means once you see it's mostly is nearly biological particle. Because fluorescence, for example, a lot of dye, a lot of organic material, they also fluorescence. So it's much more complicated. So then also we think maybe also DNA, RNA, the chain get broken and get degraded. Maybe we still can detect it. So we don't know, but they have, of course, a lot of challenge. So before us, the low one did a single particle. And because the singular even the elastic scattering is strong, but the differential singular is very weak. They said it's 10 to -3 to 10 -6 order. And so once everything you're related to the differential, you have to measure the absolute singular very precisely. So the signal very weak, very the components is element is very critical. So we did something then try to solve this problem. And traditionally, people use this setup for the detection. So generally, and use that source, then use the photoelastic modulator or the pocket cell to turn the polarization and to generate the left and the right polarization, circle polarization, that source. Then illuminate a group of particles generally sit in a substrate or in a collider, and it's like a suspension particles. And then use a detector, use a colorimeter, and rotate the detector to look at the signal from 0 degree to 180 degree. So generally, it does take a while to get a signal. So meantime, to get a signal, you need a looking amplifier to look at to match the modulator frequency and the fundamental frequency, double frequency, which frequency is matched the signal. So the system is quite complicated, and you have to get the data from a big group of particles and take a long time. So that's the disadvantage. So how to get it for single particle and for a particle run solo? So we designed this system. So first look at the very right top picture. That's the check system for the system for our setup. So when the particle we pour the air and go to the certain. Then we use a sheath nozzle to focus the particle and try to let the particle to go to our certain in land. And that stream generally is about 100 micron. And in diameter is a lambda flow. And the particle speed is about 10 meters per second. So even that, the particle is still when you're around 100 micron around or even bigger. So, but if you want, we do the optics, we always know if you want to get a very good signal, like you take a picture, you need to focus. The particle should be very exact at the focal point of the optics. So, but when you get a big collection, the depth field of the optics is always very small. So that's the tolerance is about 50 micron order. So here we use a two trigger beam and a cross beam to trigger to see the particle. So you can see here is two red beam, different wavelengths, and we cross each other and manage make them at the same height. Here is the top view of the system. You can see the two beam and cross each other, and we focus to about 50 micron diameter. The early particle pass solo the cross section of the two beam, then the two PMT, and with different filter, we can see the particle simultaneously. So once a single simultaneously detected by the two PMT, then we tell, okay, we saw a particle. Then we know the particle is at the focal point of the optics. Then we tell that recorder system to record a signal. And for the angle distribution, so we don't rotate the detector. We use an elliptical mirror. You can see here that's an elliptical mirror. We project the scattering from the particle elastic scattering at a different angle. For example, we do illuminate this way the green laser from left to the right, and the very right is 0 degree, very left is 18 degree. So all the scattering from 0 to 18 degree can project, reflect by the reflector and project to a detector. So for the image, we use the ICD, and for the phase function only, we use linear array. So here is a 32 anode PMT. So the PMT can be very sensitive and very fast response. Then we can, a few micron seconds, we can see the poor and phase function when the particle is passed solo. And for the illumination part, we look at the left top image here. So the 532 laser continually laser, and we get a clean and collimated, become a small beam. Then we make a 50-50 separation and make a two beam. One beam use a half wave plate to rotate the polarization to be perpendicular to each other. One is vertical polarized, one is horizontal polarized. Then we combine the two polarization light by ground laser polarizer. So they combine together. Then we on purpose a lot overlap each other, make them about 80 micron and separate in height and focus to a focal point around 40 micron above and 40 micron below. And you see the big image at the inside here. You can see the red laser, the dark laser for the two trigger beam is about 50 micron above the focal point. Then 40 micron above for the left circle polarization, and the right circle polarization is 40 micron below. So when the particle and get detected, then pass through the two beam, then you get two of the scattering phase function, and then you subtract and normalize them, then you get a CRDS signal. So if you use ICD, look at the scattering image from a sphere and polystyrene sphere 3 micron, you can see the images like this. So that's the forward, that's the backward, and it's the concentric and the circle. So of the scattering ring, you can see the two circles, and the corresponding angle for this pattern, for the theta is from here. You can see from 0 to 180, and the phi is like this, and is 180 at the center. So for the phase function, we just need the red box area, and from 0 to 180 in theta, and the phi is 180. And then you get the intensity distribution is like the very right and the bottom here. That's what we want. And every particle pass solo here, then we get detected. Then the two beam different circular polarization, and interact with that particle, you get two scattering phase function. Then subtract them, you get a CRDS single. So we finish the one complete measurement is about 17 micron for a complete measurement. Then we can make the whole measurement is go to the rate over 50,000 particles per second. So that's the system. And before I put the airtight box in, because I put the airtight box in, then use the pump here, I can pour the air to the system. You can see that's inlet nozzle here. That's the sheath nozzle. That's the diode laser, two diode laser here, and that's the green laser, and that's the beam, and the different polarization beam generator here. Then come to here to interact, and this one is a custom design, and elliptical reflector. Then get a lens, get the 232 analog PND, then some electronics for process the scattering amplifier and the gate and the controller, and also the single AD converter for the 32 analog PMT. So that's a full system. The size is 24 inch and 18 inch and 12 inch. So we test a lot of different particles. And mainly is non-biological particles like polystyrene, glass, and some other material, dust, and also some biological particles, but it's not the complete biological particle like with RNA, DNA, only amino acid or some other thing. And also, of course, the biological sample like the Bacillus subtilis, E. coli, and some pollen, and MS2, that's a virus similant material. So that's sample of the SEM image and generated by the eye jug. So the like a sphere is shrink by job later, then the surface and the tension make them every single particle like a sphere, but they have some different rough surface. So that's a polystyrene and the tryptophan and Bacillus subtilis. And that's the image, the pattern you get. That's the left is the forward, and right is the backward. And the dark part on the top is blocked by the nozzle. And you can see the really, and the ring, really good ring. And by the sphere, then go to the material, the surface get a roof and more roof. And then the still ring-like structure, but the get more and more and non-even distribution for the rings, and like the subtilis then get even more like islands. So that's typically the scattering pattern. So those are 3 micron long and generated by the eye jug. So then we look at every single particle, and only the PMT get the scattering phase function and the CIDS single law. And you see for the polystyrene, the red and the green and the blue, they are the two scattering phase function, but the different illumination. They are almost identical. And then you get a CIDS single law at the inside, it's almost flat, almost zero. But for the biological particle, the two picture, you can see they get a much strong single law. And if we go further and look at every single particle from the same material, Bacillus subtilis is almost 3 micron, but they have the size variation. They cannot be precisely and from the jug generated. You can see here I give and 1, 2, 3, 4, 5 different color, 5 different particles I observed. You can see there are intensity big variation from 50 to about 204 times difference. So the particle size have some difference and around 3 micron. Of course, the scattering phase packaging is big difference, but between themselves, illuminated by the two different different polarization illumination, they are close. And then you get the CRDS signal at the inside. You can see they are very close, and so they are very consistent. But of course, they still have some a lot of variation, even they are close. But the signal is much bigger than the non-biological particles. And we did a 50 particle average from different material. You can see the two line and the dark one, polystyrene and the tryptophan, they are a lot bigger signal, and all the other material from bacteria and the virus, the signal much bigger. It's at least 3 or 4 times strong. And okay, looks really promising. So then we come to a question. So now we did is all 3 micron island and generated by method. The particle generator is almost a sphere, look, and but if the particle size changes, how the signal changes, and if the particle not sphere, is the CRDS signal is related to the orientation, and the couple of the signal you see here, they are quite similar. And we don't understand. We think maybe the iJag use the electronic produce the generator, and the electronics may be oriented the particle and make the DNA, RNA, and align in the same direction. When the generator come out, then the signal get strong and similar signal. But if they are randomly distributed in the particle and the different material and the different shape, do they have the same signal, or also different growth condition for the same material? They are same. Of course, the more interesting thing we are looking is for the problem we want to overcome from the fluorescence, and is this signal is similar when they get aged or released to the air? So those questions to our mind. So then we get some more and study last 2 years, and we did some results and get some results by theoretical and experiment. And we look at this. First, you can see that's a theoretical calculation. We used a very simple model and only twisted like a spiral particle, and one twister, and I think it's 2 twister and 3 and 4 twister, and then different particle size from the bottom results. So from the low part to the upper part is from 0 to 60. That's the chi is the 2 pi a or lambda. That's the particle size. You can see the particle size increased. Is the twist effect? The twist effect is get reduced. You can see the signal at the beginning strong, get weaker and weaker for one twist, and all they have same tendency. And the blue is the negative peak, and the red is positive peak. And the x-axis is the angle from 0 to 180. And that's the scattering phase function. You can see if you cut a line horizontally, you get a phase function as what we observed. And when the particle get bigger, they get weaker. Of course, if the particle get weaker, you keep the twist density or the pitch density the same. And when the particle get bigger, then you get more twist, and maybe there should be not the result like this appear here. So anyway, this gives us some idea how the particle size changes and CRDS signal. And also we did compare with the experiment, like the same size and condition. You can see they are much quite well. And so it looks very good in some way. And we get further, and this paper just published and the end of last year. So we get a little more complicated, make the particle twisted and meet the head and the tail together. That's one twist, 5 twist, and I think that's one twist and 10, 25, and then with a lot, we say infinitive, but actually it's 100 above. So from the left to the right is the different twister, and from the top to the bottom law is the, let me see. Oh, sorry. So the first law is this one. The first column is the scattering without polarization. That's regular we see. And second is the linear polarization, and matrix 1, 2, and the last column is the mirror matrix 1, 4 minus 1, 4 all CRDS single law. So that's the single law we are interested in. And the law, first law is with the twist 1, and the second is 20, and the last law is 25. So you can see here, and the different color from the blue to the dark yellow to the light yellow is the absorption increased. You can see the absorption also affect startly of the phase function, and then the twister change, the phase function also change, and so it is not a simple results. So that means the CRDS single law is changing, and that's the disadvantage. For the fluorescence and the Raman, once the particle and the molecule is, or the particle, the material is fixed, their spectra is not changing with the angle, and not changing with the particle size orientation. But here you see the particle size and absorption definitely and affect the CRDS phase function. And also once we do single particle, the particle is following solo the system, they have a lot of uncertainty. And in order to see how the CRDS signal really like and how they change with the size and the orientation, we performed and experiment and trap the particle in the air. So you can see here, so the bottom part is the trapping. So we use a 488 and shape the beam, then use them to produce a hollow beam. So this, and why we do this way, you can see, go back to see the paper 2019 and 2014 we have to paper talk about this advantage. And chop in the air is not easy. We use this, we are successfully chop absorption and non-absorption particle in the air, and we studied it. And then the focus, use the polobatic method to focus the hollow beam to a spot to chop the particle. And at the same time, we have very long focal length, cylindrical length, to make the beam started deformed. When the beam not a circle is started formed spots, then the non-sphere particle then orientate themselves with the beam and try to, they are comfortable at the lowest potential. So then once we rotate the cylindrical length, we can change the orientation of the particle. So the particle going to rotate, follow the rotation of the cylindrical length. So we use a step model control the cylindrical length orientation. Then we can study how the CRDS single law change with the particle orientation, also the size. So use the optic chip and orientation controlled set up, we can and really and precise study the CRDS function or scattering function of the particles. So here is some results, and this just submitted to GQSIT the paper, and you can see at the beginning is some long CRDS intensity get strong, and different orientation and phase function different. It's quite complicated, and so, but whatever is whatever. That's the results we observed, and so low data, and we did for single particle. We did the theory calculation. We did the study from trapped particle or the flow solar particle. We get some results, and hope this really can be useful for the bioaerosol detection, for the faster detection in the future. And but the more interesting thing we wanted to see is how the particle interact with the atmosphere air, how the property change for the sphere. And that's a paper just published. Here said it's submitted. Actually, this paper is already posted in the web. Is aerosol science, a journal of aerosol science. So we generated the particle and use a sound taker, and then release to the chamber, reaction chamber, and the chamber we can put a UV exposure to the chamber, and also change the ozone and ozone concentration, and also the humidity of the reaction chamber. Then we pour the particle to measure the particle fluorescence by the Weber's, by the macro droplet measurement company, the Weber's system, and that can measure the visible band by UV and blue excitation, flash lamp. Also, by the deep UV excitation, you measure the UV band that can measure the total intensity of the 3 bands of the fluorescence. And at the same time, we measure the single particle CRDS singular phase function, and pour from the chamber at a different time. Then we look at the results. So you can see, and that's the fluorescence about 2-3 micron. That's the 2-8 excitation, the UV bands, 2-8 excitation, and the visible bands. Then the very light is the blue or UV stare, UV excitation, and the visible bands. So you can see from the 3 condition, that's on the humidity 60%, and without put the UV and the ozone on. So put the UV on, put the ozone on, the deep UV band, dropped greatly, and the visible get some drop, not too much. Look at the score of the intensity. They are different. And the visible one also dropped a little, and surprisingly, the CRDS phase function, they are very similar at the 3 condition. So that's very encouraging to us. And so it's only we test the MS2 particles, and we don't know how they really and how this happened, and we need more study. And go to summary. We developed a new system, and that can measure the CRDS phase function from individual flow solar particle, and without the early moving and parts and the modulator or local amplifier, and make the system very simple, very sensitive, and can measure more than 50 particles per second. And we can see the particle how DNA, RNA, and they have strong CRDS signal compared to the non-biological particles. And the complicated part is the CRDS phase function or profile and the intensity, they change with the particle size orientation, and also maybe growth condition. But very encouraging is their how small and influence for the environmental condition or aging process. So they are similar for the one single test, and that seems better than the fluorescence. Okay, go to the end, and I thank you for the funding support and all the other people work together with this project at the AIR. We have the young people, Amble and UD, and my colleague Gordon also. Then the Professor Chu Wang's group at Mississippi State University, and his student Rafael, and George Santapia, and at the University of Nebraska Medical Center, and his student, and the last experiment was done at his lab, and eventually master term is going to send to his lab for BSL 1 or 2, and or even more complicated measurement. So then the theoretical calculation is mainly performed by the two professors from Ukraine, and they are actually is in Germany and Poland. Here they are did the calculation with us. And thank you for your attention. Okay, I'm done.
Thank you, Dr. Ban. Wonderful talk. I guess for now we can transition into a little short Q&A session. We have a couple questions in the chat. The first one is from Tom. It's, how sensitive is the CIDS technique applied to identifying bioaerosols to atmospheric gas phase composition, e.g. ozone, water vapor?
Percentage don't know. So suppose there should be have some change. And actually, and I read a paper recently published last 2 years ago, and they look at the single cell. Of course, that cell is on a microscope layer on the substrate. Then you can have a long time to look at the cell. So that when the cell is changing with some buffer liquid, the property change, the CRDS can see the change. So it's really sensitive. Too sensitive, then make it trouble for really fast detection. And you're supposed to see the difference from different kind of particle or different condition. But if, like our system, the particle is non-solid system, then it's harder to tell the difference is from the property change or from the particle position change or size change. It is very tough. So we are still in the early stage of this technology. So we don't know how good they are in somehow. Is that the answer for the question?
Second question was by Reinhard. Excuse me if I mispronounce this. Reinhard Niesner. Is CIDS effective for glycerol encapsulated bacteria? The scattering will primarily be influenced by the coating.
Don't know. I don't know. Honestly, you see the results. Have some good result. Good is beginning, and the other people did only a fair sample. People did from the group of bacteria. Is the use a fish tail cell something, stem cell. That's big. I guess that signal is strong. That's the results. And recently, the people did is use the microscope and did something, but I don't know honestly.
Okay. Another question from Tom was, when will the CIDS measurement system be ready for use in the field?
So now is I try to deliver, and actually supposed to deliver last year. Then something happened is the order and something. Then now is the alignment is finished. Now you see the picture, but I didn't put the airtight box on yet. When I put the tight box on, then I have to test when the pour the air in and load the aerosol generator and the system how I can make it work, and that should be shortly. So that is kind of like is can be used in the field if lucky enough. Should be 1 month, 2 months. I'm going to ship it to Josh and let him play. He planned to do in the BSL1, BSL2 lab, and also to do the measurement in outside field.
And then we have one final question, unless anyone else has any, from Peter Hairston, which is, has signal processing been implemented that enables classification of an unknown aerosol? And he says, thank you for the talk.
No, not totally understand the question. So he asked that when I do the subtraction and the normalization is early special. So can you repeat the question again? Sorry.
Yeah. So the question was, has signal processing been implemented that enables classification of an unknown aerosol? If Peter would also like to unmute and kind of bolster his question, feel free to do so.
I still not get it. So the non-bio, I guess non-bio, I look at the singularly centered we get from the chopping particle. And it's kind of complicated. For the sphere and non-biological and uniform sphere like a glass bean, polystyrene sphere, even the biological molecule from tryptophan, they are the CRDS singular is very weak. They are almost zero, or they are uniformly is like a kind of noise around the 0 to 180 degree. So it's like a randomly and positive, negative, and weak. And for the biological, it's much strong. That's the basic and results we get a lot. But unfortunately, and some particle like the horizontal load dust, and the singular also, some of them quite big. So we don't understand. Then we think maybe is that horizontal load dust is not a sphere, and also the dust maybe have some content of bioresonance inside. So that's we talk in the paper and just submitted to the JKSIT. And so, yeah, it's still don't completely understand yet something. We just kind of like this is, we last a couple of years, we doing this, focus on this topic. And that's all the results you'll see here. And I don't know if I answered the question or not.
I mean, just to clarify in his question, he said, can you do blind testing with automatic classification?
That's what we want. Yeah. So law is, yeah, that's always a good question. Always the problem. You know, a lot of people say, oh, we can do this, do that. Actually, they just use the laboratory sample, the load sample, a field sample. They say, oh, we can do this, do that. But actually, once you do the blind test and run the real experiment in the air, that maybe doesn't work. So we don't know. We that's what we wanted to do. So the next step, we're going to do more in the lab. Also, at the same time, we are going to do in the air. So obviously, in the air, they have a lot of biological particles itself. So what's the threshold to put it there? What's the percentage of there? So we need to test. We use it. That's the next step. Definitely, it's a good question. It's really we need to figure out.
Okay. Do we have any other questions? I'll give everyone a minute to, if you have any final questions.
Hi, I have a question. Hi, Yongle. That was a good talk. Thank you.
Thank you.
Hey, I had a question about the bioaerosol reaction chamber. How much time do the particles spend in that chamber while you're challenging them?
Actually, I don't have the answer here. I need to look back to the paper. Definitely, they have a paper published, talk about the chamber, published or at least submitted. I don't know, published a lot. And this paper, and it's already posted on the Journal of Aerosol Science. The first author is Daniel, and Daniel Ackerman is the Josh student. In that paper, we started that paper of the reaction chamber. So it's new. I don't know how long. Before we work together, is use the rotation drum. That one you can couple hours is reasonably okay. And we did a measurement every 15 minutes or 30 minutes. And we can measure about a couple hours, 4 hours per day and 2 experiments. That's what we did. But for this new chamber, and I didn't do the experiment together. I went to their lab and show them how to use the CRDS measurement system. So I have a system there before. It's not the way I show in the picture. Then the student did an experiment, and I don't know how the chamber, the particle can stay there for like the circle of time generally. So before it's like go to E, 1 over E, and it can be couple hours. And I don't know this one. Of course, it depends on the particle size also. Small particle, you can last longer. And but the results come out, I think it's a half-hour results.
Okay, yeah, that's great. I'll look forward to that paper and look at it. Thank you.
Sure, thank you.
All right. Do we have any other questions?
Can email me, but mostly I maybe cannot answer. And you see all the results is on the slide, and your guy can get it from the Chopper Air website. And if you have anything wrong, please correct me, and yeah, send me email also.
All right. If we have no more questions, I just want to once again thank Dr. Pan for this wonderful talk. It was very interesting. And yeah.
Okay. See you guys in Chopper Air.
Yep.
Buffalo.